mostly crowded my last video was on gluconeogenesis, I just wanted to make two separate videos talking about really the first two enzymes, pyruvate carboxylase, and then I'll make another video for phosphoenol pyruvate carboxykinase, because I didn't think I had much time to talk about the mechanisms of their actions and how they're regulated and things like that. So the reaction that we're talking about is ATP and pyruvate acting with carboxylate group, or CO2, dissolved in water through the use of pyruvate carboxylase is going to give us, we're going to be going from a 3-carbon sugar, we're going to be adding that to it, we're going to be making oxaloacetate, and then obviously some of the just byproducts of ADP and inorganic phosphate. So what I had wanted to mention about pyruvate carboxylase that I didn't really have, the room or the time to talk about in the gluconeogenesis video, is that this is a mitochondrial enzyme. So one of the things that we have here is compartmentalization, and this is what you're going to find out is that compartmentalization is not just nice in terms of keeping things tidy in the cell, but in terms of solving the issue of making cells more energy efficient to meet a metabolic demand that we have of being eukaryotes as opposed to prokaryotes. It makes regulation so much easier, right? So mitochondrial enzymes, this is all happening in the mitochondria. In this context, and if you're alpha proteobacteria, well, you know where that story comes from. But anyways, the cofactor for pyruvate carboxylase is biotin. Not forgetting our, I guess, general things that we talked about at the very beginning, but this is covalently linked via an amide bond with lysine. And so that may kind of seem familiar as to you, but with lysine, it's very long. And so it's a very long, flexible chain that gives us kind of some room to kind of fling it around like a flail, if you wanted to think about it, for doing these reactions. Other thing that I wanted to mention about biotin is that it's involved in the carrying, or you could think of it as transferring, of activated CO2. And what I mean by the words activated is that it has been phosphorylated. This is actually why we use ATP in this reaction. Last thing I wanted to say about the pyruvate carboxylase, and then I'll be done with this video, is that acetyl-CoA is an allosteric activator. So as you can imagine, if it's an allosteric activator, once acetyl-CoA binds to its allosteric site, it's going to undergo a conformational change that enables biotin to do his job, that enables the reaction to happen, right? But this biotin can't work. Biotin cannot be involved in transferring that CO2 from one area to the next unless acetyl-CoA is bound to it. And by the same notion, the concentration of acetyl-CoA plays a role in the rate at which pyruvate carboxylase